Okay, so what we want to do now is make a palette which is essentially devised for SPGen. So we have all of these palettes here. We've seen we have different pictures, different things in there. And what we want to do is make a palette of tools that or symbols that we can use for drawing pathways, because obviously all of these palettes are not something that we will use in drawing pathway diagrams. If I was drawing a computer network or a social interaction network, I could possibly use the symbols that are already there. For what we want to do something else. Okay, so have we still got the file from yesterday? Okay, so if we go into that file, and I'm going to go on here, I'll show you which one it is. I've got to find out which one it is first. So under sample data, under MEPN pathways, you'll see a little folder saying notation. And in there is this palette. So if we go here, if I click on here, just to show you, we have the notation system that we defined. Okay, so slightly outdated this one, but don't worry about that. So we're going to create in here a custom palette, and that palette is going to be all the shapes that we need to draw the diagrams that we're going to look at in a moment. So I'll do this once, and then you can hopefully go. So we're going to manage palette. And so we're going to a new section, and we're going to call this MEPN. Okay. And we're going to import. Is it import symbols? Sorry, it's been a while since I've done this, so I'm going to have to go onto here, find that file that says sample data, MEPN pathways, notation, and the one that says MEPN verse 2. I'm hoping that will work. Okay, so we've got it there. And hopefully now it's also in here. Okay, so now he said hopefully we have a series of shapes in there which are the palette that we will use in drawing diagram. Right, so yeah, sorry, I was we're actually importing a palette, so let me just delete that. So I'll delete it again and go through it again. So what we're actually doing here is creating a palette of shapes that we can use in constructing pathways, and it means that now you'll just be able to drag the appropriate shape. When you want to draw a protein, you'll just be able to drag it from the right hand side, and you'll have a protein represented on the screen. So shall I go through this again? So if we go here to edit and manage palette, which is near the bottom, and we're going to import section, and in this folder called notation, which is in the sample data MEPN pathways notation, we will find this file here called MEPN palette, and we say open. And what we should then have in the right hand side is all the symbols that we need to draw an MEPN diagram. So in here now we have the shapes, so let me just close that down. Have we all got the palette in there? I'm just going to close that and do a new window. Okay, so now what you will see on the right hand side, let's just close that one and open that one, is that if we wanted to draw a complex, we could just drag that over and we'd have the shape and the color of a complex. If we wanted to draw a protein, we'd draw that over here, etc., etc. Okay? Does that make sense? So what I'm going to try and do now is I just want to, I guess what would be useful for you to do is open the actual notation system as a separate file. So go into MEPN pathways and go into notation. Don't worry, it's called, this notation is called, well, I was going to rename the notation system graphical computational modeling language. I've decided to go against that, but that's what's the file still in there. And what you'll have is this version here, yeah? Okay. So if you want to go back and look at what a given type of entity should be represented as, this is your key here to do so. And you can sort of understand what shape or color you might do. Now this is an exercise that we're going to go through now, which is designed to show you actually that you all don't think very logically. Okay, I mean that's a bit harsh, but I mean basically what I'm going to do is show you a bit of text. I'm going to give you the symbols of the things that are talked about in that text, and I'm going to ask you to try and draw it, okay, using this idea that we've got proteins that interact with each other. I've used a very relatively concise piece of language to describe a certain set of events. I'm going to give you the parts, and then I'm going to ask you to try and draw those parts, give you 20 minutes or so, and then perhaps we'll finish there, and we'll have a look after the break of how some of you got on and had a look. See, so some of you might get there. I'd be delighted if you did, but quite often you'll find this quite a difficult exercise to do. So myself and Laura will be wandering around and helping you with it, but obviously we don't want to directly show it. Part of the process of this is to show that it isn't that obvious how you put things together as a visual language of events. Okay, so I'm going to ask you to open another file now. Let's go up from notation, and I'm going to ask you to open this interferon beta parts.graphml. And what you should get is this here. And what I'm drawing here is a series of components, and what I'm asking you to do is to stitch them together so that they represent the text that I'm going to show. So these are the bits that I think you need to draw a bit of biology, and that biology I'll show you as a piece of text which has been written in a relatively concise format that will allow you to then string these together so that you end up with a diagram that represents the information in the text. Does that make sense? Happy with that idea? Say, maybe you'll be able to get there, and I'll be delighted if you can. So you've got the parts list. Now here's the kind of sentence that we want to do. So I would like you to look at that text. And so this is a cytokine called interferon beta, and this describes the signaling pathway from the interferon interacting with its receptor to the downstream events of what happens then to the point where interferon activates a number of genes downstream of that. Okay, so there's a interaction between the ligand and the receptor. There's a secondary signaling system whereby that interaction is transported through the cytoplasm to the nucleus, and transcription factors activated that then activate certain genes. Okay, so what I'd like you to do is look at that text, look at the symbols that I've given you in those parts lists, and try and draw what we've seen with this idea that I've been trying to communicate with you about how we can draw diagrams as a series of events using arrows, nodes, components, processes, etc.